I have you loud and clear.
Hello. Welcome. Science.
And that is to say,
physics,
medicine,
nature,
or
Space. Time.
the brain,
Life.
the universe.
Hello! This week, the tiny molecular chemists that make the world go round. Catalysts, from brewing beer to cleaning up car emissions and even making less polluting fuels. We're asking, what exactly are catalysts and how do they work?
Plus, scientists discover the mechanism behind the majority of Alzheimer's cases, new technology helps beekeepers keep bees, and we explore the prospects for the survival of humanity. No biggie. I'm Izzy Clark.
I'm Katie Haylor, and this is The Naked Scientists.
The Naked Scientists podcast is powered by ukfast.co.uk.
First up, neurodegenerative diseases like Alzheimer's are incurable conditions which cause the degeneration or death of cells in the brain and can result in deterioration of movement, memory, and cognition. Scientists are desperately searching for a cure, but we still don't know what causes it in the majority of cases. Now, a team at the University of Cambridge have used a new technique to discover the mechanism behind the development of these diseases. Georgia Mills spoke to Patrick Chinnery, Professor of Neurology at the University of Cambridge.
Late-onset neurodegenerative diseases like Alzheimer's disease are on the increase because as the population gets older, more and more people are suffering from these disorders. And the truth is, we don't know the cause in the majority of cases. So we were keen to try and work out whether or not some new genetic mechanisms might be responsible for some of these problems. In 95% of cases, there's no family history, and we don't quite understand the cause in those patients. It might be partly environment, and it might be partly some genetic risk factors, and we were keen to explore a new genetic mechanism by studying the brains that we've collected from patients who've died from these conditions.
Right, so how did you investigate these brains?
We took advantage of a resource that the Medical Research Council has been investing in for decades, actually. These are brains that people have donated for medical science and research, and we identified about 50 of these brains, partly from people affected by Alzheimer's disease and another kind of dementia, and also some healthy brains that had been given by people who died from, for example, a road traffic accident who weren't suffering from the condition. And we developed a new technique that allowed us to look at the genetic code in extreme detail across bits of the genetic code that we know can cause Alzheimer's disease and related conditions. And what we found is that almost all of these brains were containing spelling mistakes in the genetic code, affecting some of the cells in the brain. We found these all over the brain when we looked, but not all the cells in the brain were affected. Only a small proportion of the cells were affected. In fact, the technique we developed for the first time allowed us to do this on brain tissue.
Right, could you tell me a bit about that technique?
Essentially what we do is we sequence a particular bit of the genetic code, the DNA, many times over. And in this study, we sequenced it 5,000 times in each sample of the brain that we looked at. And that allows you to look for very rare genetic changes that you wouldn't detect if you only sequenced it perhaps 2 or 3 times.
What has this told us, the fact that these spelling mistakes are appearing in certain cells but not all of them? What can we take from this?
Well, what we've learned from this is that contrary to what people previously thought, which is that the genetic code in your brain and my brain is all identical, actually there's a lot of heterogeneity. A word we'd use to describe that is a mosaic. So there are all sorts of different genetic variants in the cells in our brain. Now, some of those might be completely innocent and not cause any problems, but by measuring the frequency with which they occur, we can make some predictions about how likely it is that they'd affect a bit of the genetic code that could predispose to a disease in one of the cells. And if that happened to enough cells, it could contribute to something like Alzheimer's disease.
Right, so when do we think these mistakes are happening then?
The thing about the brain is that the cells generally don't divide, so they're not being copied because they're set in place when the brain develops in the womb. And the pattern of the genetic change we saw suggested that actually the spelling mistakes had occurred when the brain was being formed as the cells were being copied and divided to grow from a simple embryo, which is a ball of cells, into something more complicated with the brain structure itself.
Right, and then this would explain why some areas of the brain are slightly different if these mistakes happened when some of the brain had already sort of formed.
So you've hit the nail on the head, exactly. If you could imagine, in very simple terms, ultimately the brain derived from a cluster of cells. When we're actually fully formed, We have 100 billion cells in our brain. And so you can imagine this as a branching tree-like process as the cells divide and form more and more daughter cells. And depending on when the spelling mistake occurred would determine how many of the cells were affected. If it happened really early, most of the cells would be affected. If it happened really late, only a few of the cells would be affected. And what we saw in our experiment was that all these possibilities were occurring.
Now we've discovered this mechanism. Is there anything we can take forward from this that might give us insights into one day treating it?
I'm sat here talking to you now, and I know from this work we've done that I'll have spelling mistakes in different bits of genetic code in my brain. It doesn't really change the fact that those were there before we discovered this was happening. So I don't think it has a major impact now on how we would diagnose or treat people. But by getting these insights, I think it's given us a clue as to mechanisms. And I think that's important because it may be that using an adapted version of this technique, we might be able to help diagnose these conditions at an earlier stage. That's really important because I think the field generally agrees that the sooner we intervene with treatments, the more likely they are to be effective. And by understanding the mechanism, of course, we can start to develop treatments targeting that particular mechanism, the protein that's altered by the genetic change, and thereby move to a position where we've actually got effective treatments, which unfortunately are not there at the moment.
And hopefully this will take us one step closer. Patrick Chinnery there from the University of Cambridge speaking to Georgia Mills, and that study is out this week in Nature Communications.
It's certainly important work, isn't it? Now, how can you plan for the future? No, we're not talking about your savings or your personal 5-year or even 10-year plan. We're talking about the future of humanity. Technology, society, and even the climate seem to be changing at unprecedented rates. So how do you prepare for an uncertain future? This tricky topic is being tackled in a new book called On the Future by Professor Lord Martin Rees, who's the Astronomer Royal, and he joins us now. Welcome, Martin. Now, how far into the future can we try and plan?
Well, of course, we can't predict on Earth as confidently as in the heavens, but I think there are a couple of things we can predict at least 50 years ahead. One is that the world will get more crowded. The world population is going up, mainly because people are living longer and because the birth rate is high in Africa and parts of India. So we can predict that the world population by 2050 will be about 9 billion, and that means we'll have more people to feed and they'll be more demanding of energy and resources. And that leads to a second prediction, which is that because of all those things, it's almost certain the world will be warmer, because the carbon dioxide concentration, which is going up, will almost certainly continue to go up. And there's a big time lag in its effect, so almost certainly we'll have to contend with a changed and warmer climate.
Absolutely. So are those sort of our major threats sort of to our natural planets, essentially?
Well, that's right. Of course, because I'm an astronomer, people think, do I worry about asteroid impact, etc.? They can happen, but they're rare. What is worrying about the two things I mentioned is, as you said in your introduction, the rate at which things are changing because of expanding populations and because of advancing technology.
And in your book, you also mention the report of the Intergovernmental Panel on Climate Change, who recently announced their recent report. So how important is this?
Well, I think there's a risk that if the temperature rises, we might cross so-called tipping points when some really serious effect happens. And we've got to think not just about 50 years, but about what will happen at the beginning of the next century, because any baby born now is quite likely to be alive in the early part of the 22nd century. So we've got to think about how we can pay an insurance premium now to reduce a risk which otherwise will be encountered by those who are recently born. I would say that if we want to deal with climate change, then in my view the only effective way will be new technology. I'm rather sceptical about whether politicians will agree to cut carbon for the benefit of people 50 years in the future in remote parts of the world. But I'm slightly less pessimistic now, and I do think we do have a future. And what I say in this book is that the best solution to the climate problem is to accelerate research and development into all forms of clean energy. Because if we can have carbon-free energy which is developed fast and cheap, then countries like India, which certainly need to have more power, they will not be able to afford to have anything other than coal-fired power stations unless the cost of renewable energy comes down. So if it comes down, they can leapfrog directly to clean energy. That's good for them and good for the country which develops the technology.
Absolutely. So if we can get that technology, a lot of us are living our lives online. I mean, is there any chance that even that could take over and perhaps go in a negative direction?
Well, it's a worry because I said we could predict some things 50 years ahead. But what we can't predict is technology because smartphones and the internet would have seemed magic even just 30 years ago. So if we think about 2050 and beyond, we can't be confident of whether there'll be some new breakthrough. And in particular, I discuss in my book two technologies, cyber and bio, which offer huge benefits, but also they present serious risks. And the reason they do that is that it's possible for just a few people, by error or by design, to have an effect that cascades globally. We know about cyber attacks and how catastrophic they can be, and we're going to be more vulnerable in that way. And also possible misuse of biotech can happen in the same way. So I think we need to be concerned about those two technologies.
And when we talk about biotech, what do we mean? What does that encompass?
Well, of course, we know we will have gene sequencing and maybe gene synthesis, which will allow us to do certain things which you may feel are ethically or prudentially not a good idea to do. But also we have the risk that there will be some experiments that go wrong. About 5 years ago, people did experiments to show that you could make the flu virus more virulent and more transmissible. And the American government stopped funding these experiments because obviously they're risky. And there are other experiments like that. And whereas 50 years ago, when molecular biology started, there were just a few labs and they could regulate themselves. Now these technologies are global, there are strong commercial pressures, and I really worry because we want to try and regulate them, but regulation can't be enforced globally, in my view, effectively because of these pressures, any more than the drug laws or the tax laws can. And if you ask me what keeps me awake at night, it is the misuse of cybertech and biotech.
Okay, so very quickly, what do you think we need to do to make sure the future of our humanity has a positive outcome? Because the book itself is quite optimistic.
Well, that's right. I think there's a big gap between the way things could be and the way things are, and technology has a huge upside, but to go with that there are risks. So I think awareness of these things and ensure that the benefits of these technologies don't just go to an elite, but are widely spread, and in particular that they go to the countries like Africa, where otherwise the population may run away and there may be real pressures.
Professor Lord Martin Rees, thank you very much. And On the Future is out this week in hard copy, and you can also get it as an audiobook.
So what we were trying to do with this paper is to demonstrate the ability to type using brain signals, anywhere between approximately 4 and approximately 8 words per minute, a factor of between 2 and 4 faster than what's been demonstrated before.
Each month, the eLife podcast talks to some of the world's best scientists. Join me, Chris Smith, as I hear about breathtaking discoveries hot off the press. Find the eLife podcast on iTunes or listen and download for free from nakedscientist.com/elife.
Still to come, the latest mission to Mercury, and we meet the mini chemists that live inside your car, your beer, and your mouth. But first, it's time for this week's myth conception. And it's a myth that's been making Tamsin Bell's blood boil.
I remember at school, a friend told me that blood is blue until it is exposed to air, when it turns red. Looking down at my wrists and seeing those blue lines, I had no reason to doubt them. But like almost everything you hear in the playground, it was complete nonsense. Human blood is the same colour whether it's inside or outside your body, and that's red. The colour is brighter when blood is full of oxygen and darker red when it is oxygen poor, but it's definitely not blue. So how did this myth start? It may come from the fact that the veins we can see on our body are used to carry oxygen-poor blood, and these look blue. And to add to the confusion of children everywhere, the typical blood flow diagram of the body taught at school uses red lines to represent oxygen-rich blood, whilst blue lines correspond to oxygen-poor blood. To understand why this myth is false, let's consider the science behind this red substance. Blood is our internal delivery system. It takes nutrients and oxygen all around the body via super speedy highways known as arteries, and then takes waste products like carbon dioxide away through the veins. Our blood is made up of cells floating in a liquid called plasma. Blood plasma is predominantly composed of water, but also contains proteins, sugars, ions, hormones, and carbon dioxide. The cells we have in our blood include the little donut-shaped red blood cells which carry oxygen, white blood cells which fight infection, and platelets which help the blood to clot, stopping it from endlessly pouring out of our bodies after a cut. Interestingly, our blood also contains small amounts of some metals, even expensive ones like gold. The colour of blood is determined by the protein in the blood that carries oxygen around. In vertebrates like us, the colour comes from a protein called hemoglobin. This protein contains iron, which makes blood bright red when it's stuck to oxygen and dark red without oxygen. Different proteins lead to different blood colours. For example, cut horseshoe crabs and they bleed blue, while sea squirts, disgustingly, have a mustard yellow coloured blood. Other weird and wonderful sea creatures have fabulously vibrant blood colours such as green, purple and pink. So why does our red human blood look blue in veins close to the surface of our skin? Well, the size of the blood channel, the oxygen content of the blood, and the thickness of the skin above the channel affects how visible light travels through skin, which is known as absorption. And these aspects also affect how much light can escape or be scattered, changing the colour we see. The oxygen-poor blood in our veins is best at absorbing red light, but blue light cannot reach deep into the skin. So a shallow vein appears blue because this colour is reflected back at the eye. We cannot see our arteries because they tend to be smaller and have thicker walls than our veins. Hopefully this misconception was not in vain, and you'll remember that, unless you're a sea squirt, our blood is always red.
It certainly wasn't in vain, thanks Tamsin. And if you've heard a biological bluster or a physics fairy tale and you'd like us to take a look, you can email it to chris@thenakedscientists.com.
I love that. Imagine having yellow blood. Ooh. Now, Katie, did you hear about this space launch on Saturday, the 20th of October?
I did, and I'm hoping you're going to tell me more.
I certainly can. So, only 2 missions have ever tried to explore the planet closest to our sun. That's Mercury. First in 1973 with the Mariner 10 mission. Then there was Messenger reaching the planet in 2015. Now, on Saturday, the 20th of October, the European Space Agency and the Japan Aerospace Exploration Agency launched a new spacecraft to Mercury. But why go back, and what does this new mission hope to learn? I spoke to David Rothery from the Open University, who's involved in the mission.
Mercury is 3 times closer to the Sun than the Earth, and it's the smallest planet in the solar system. It's a very dense rocky body, which means its core of mainly iron is much bigger compared to its overall size than in the case of the Earth. And it was thought to be quite a dull, boring place because of this very thin rocky shell. But it turns out that the rocky shell is full of sort of active ingredients. It's a volatile, rich planet. The whole surface has been made by a series of big volcanic eruptions, mostly lava flows. But in the more recent past, the eruptions have been explosive, which is a surprise. Because to have an explosive eruption, you've got to have gas involved in the magma. So we're now mystified. Did Mercury form there and somehow retain its volatiles? Very hard to understand how that could happen. Or did Mercury form further from the Sun and migrate inwards as part of the birth of the solar system?
To answer these big questions, the European Space Agency and the Japanese Aerospace Exploration Agency are sending a twinned spacecraft called Bepi Colombo to explore this hostile planet.
One of the aims of the mission is to understand the planet's magnetic field. So seeing the same thing from 2 different perspectives is really going to reveal what's going on in the magnetic field.
Another part of the mission is to explore what Mercury's surface actually looks like.
My role is I'm leading the European Space Agency's Surface and Composition Working Group for the planet, and that suits me as a geologist. I want to understand the surface history and what it's made of. I'm also on the team for the British-built instrument, which is the Mercury Imaging X-ray Spectrometer, or MIX, led from Leicester University, and that will determine the elemental abundances across the surface. We know there's a lot of sulphur. We don't know the distribution of the sulphur. We think it's probably depleted where there have been volcanic explosions, but we don't know for sure. That's one thing we hope to find out.
But travelling to Mercury, the closest planet to our Sun, is no easy task.
Oh, I'm very excited. On the past, we had some problems, we had some hiccups, because it's a very challenging mission with a challenging design.
That's ESA's Johannes Benkoff. He's the project scientist of the Bepi-Colombo mission.
The problem is when you want to send a spacecraft to Mercury, it's not the distance. You need a lot of energy to go in the direction of the Sun. For that reason, we have built our mission on a solar electric propulsion system, which we use in order to bring our spacecraft to Mercury. And this solar electric propulsion engine is very energy efficient, so that we have to use less propellant than when we would use a conventional propellant in order to bring our spacecraft there.
Having left the Earth, Bepi-Colombo will come back past our planet, which will slingshot Bepi-Colombo around Venus twice, and then complete 6 flybys of Mercury. On the final approach, the spacecraft will have slowed down enough to glide into the planet's orbit. But why not just fly directly?
You would need much more energy or much more propellant to go there directly, which we unfortunately cannot design a spacecraft for, because we have no rocket which then will bring such a heavy spacecraft and fly it directly to Mercury.
Once orbiting, the two spacecrafts will separate. The Japanese craft will stay in orbit to explore Mercury's magnetic field, but the European craft will nudge a bit closer to get a good look at the planet's surface. Another challenge is that the twinned spacecraft will need to tolerate Mercury's fiery hot neighbour, our Sun.
For the heat, we had to develop special insulation blankets in order to protect our spacecraft, because on the outside we have the heat from the Sun, but also the infrared re-radiation from the planet itself, which makes it very complicated to keep the inside of the spacecraft cool. So we have a special insulation blanket which is several centimeters thick and contains ceramic, textile outer layer, and then we have titanium and aluminium layers. And at the end we are able to have on the outside temperatures around 450 degrees, and in the inside we have almost room temperatures to operate our instruments.
Johannes has been working on this project for the last 15 years, and now Bepi-Colombo is finally launching. So how long until we get that all-important data? Back to David Rothery.
It will take 7 and a bit years to get to Mercury. We'll get there round about the end of 2025. We'll be in a weakly captured orbit and have to optimise that, and it won't be till the spring of 2026 that we start our science operations. The spacecraft has a design life of a year. If it survives in Mercury for 1 Earth year, the mission will be a success. If it's still healthy, we hope to go for a 2nd Earth year. NASA's mission Messenger managed 4 years at Mercury, which would delight us if we can do it.
Fingers crossed, just a 7-year wait to find out. That was David Rothery from the Open University, and before him, Johannes Benkoff from the European Space Agency.
And finally, a little closer to home, bees. Bees are critical to our continued existence. They pollinate our food, taking pollen from one plant to another, allowing them to reproduce. And without bees, humanity would, quite frankly, begin to starve. So anything that helps them is welcome. Now, a company called Oracle Technology, in conjunction with the World Bee Project, have created a system that pools the data of sensors into one big, accurate database, keeping beekeepers up to date. Talk about hive mind. Adam Murphy spoke to John Abel, Vice President of Cloud Technology for Oracle UK, Ireland and Israel. As it turns out, we have a lot to thank bees for.
If you look at the world we have today, the critical element is food for the growing population. 1/3 of our food is actually down to pollination, and the pollinators The honeybees is one of the key resources that do pollination. The problem is, in England alone, between 1985 and 2005, there's been a 54% reduction in those pollinators.
And could you tell us then about the tech you've developed to help them out?
So on our hives that we have here at Oracle Reading, we have the ability to capture sound, we have the ability to capture humidity, the weight of the hive, and also we can look at the external factors like weather and rain. What this means is the ecosystem of the hive, and specifically sound, which is the exciting one, we're capturing and putting into the Oracle Cloud. If you think about it, 2 hives give us a good set of data, but more and more hives that add on top of the World Bee Project hive network allow us to grow an ecosystem where we start seeing en masse how hives are happy or unhappy depending on the environment they live in. So we can start seeing very quickly how to inform the beekeeper of where to place the hives for the best impact, to yield great honey, but also to have great pollination.
How do we get from the data you have to actually helping the beekeeper? How do we apply the results that these sensors give us?
What happens in the first instance is we can give scientists data en masse, and using this new technology, we can start looking for patterns in the data. So we could start actually indicating to multiple beekeepers, is there a threat in your area? So it could be that there's a hive in the area that's been attacked by Asian hornets. So it'd be good to inform other beekeepers that's happening somewhere else so they can be ready to react. Maybe there's a disease. So again, informing other beekeepers there's a disease means they're ready to act. We can intelligently tell the beekeeper about what's happening around them or around the hives in their area. We can also tell them about their hive if someone is, let's say, lifting the lid when they're not there, because the weight sensor will have a drop of weight. We can tell from the sound if the bees are going to swarm, which means they could leave the hive. This can instantly be passed to the beekeeper. And also, when people do building applications for new blocks of offices like the one I'm standing in at the moment, in the future we can give them what's the ecosystem that will benefit pollination. So there's huge ramifications for this data to be shared and used for growing our food resources, protecting the bees, and making our landscape a lot more eco-friendly.
Does this sensor, when it goes onto the hive, does it bother the bees at all? Is it intrusive?
I mean, it's the size of a matchstick, if you think at the end of it. So it's very small. The bees don't really know what it is. It's so in the corner.
And is it expensive, this system, or can anyone feasibly get it for their beehive?
Compared to the cost of the hive, they're actually a lower cost than you think. And actually, we hope as people get more and more into this scheme that they start adding their data onto the Oracle Cloud, that it will come down in unit price. One of the other things that I have a personal vision for is, wouldn't it be fantastic if schools had the ability to create their own Raspberry Pis or microbits, and they worked with their local beekeepers to put their own sensors on there and add their own devices. A lot of these are very low cost to acquire. You could build it with a small amount of knowledge, and that would be a great way to collaborate, and obviously people being educated into why pollination and the pollinators are so important.
That was John Abel speaking to Adam Murphy about their unbelievable work with the World Bee Project.
Oh dear. And if you would like to find out more about any of the news we've just covered, all the transcripts and the papers themselves can be found on our website, thenakedscientist.com.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
This week, we're talking about the tiny molecular machines that we may not be aware of, but we can't live without. These are called catalysts. For me, this word brings shudders and flashbacks to chemistry lessons at school.
Well, don't worry, Katie, because during the show, we're going to tickle your scientific taste buds and catalyse your interest into these impressive microscopic chemists which are so vital for life. So perhaps let's start with a refresher on our chemistry lessons. How do catalysts work and what do they have to do with beer? Liliana Fruck from Cambridge University is in the studio with us to tell us about them. So Liliana, what exactly is a catalyst?
everywhere around us and also in our body, there are continuous chemical reaction happening. That means one compound is transformed into another compound. And this one sometimes goes fast, sometimes slow, and requires energy and special conditions. And catalysts are actually compounds that help these reactions happen and go faster and more efficient.
Okay, so how do they work exactly?
So, for example, in nature, we have catalysts, and they're called enzymes. They are made of smaller elements. They belong to the class of proteins, and they basically lower something which is called the activation energy of a reaction. That means every reaction, like every two people in a party, they need a certain activation energy to talk to each other. Compounds need this activation energy as well. And enzymes are mediators. They lower this activation energy so that compounds can come together.
So where do we get these enzymes?
So, for example, we have them in our mouth. We have them in our body. They are everywhere in the nature. So if you, for example, had some potato or pasta for lunch today, the first thing that would happen when you put them in your mouth, there will be an enzyme that will degrade the large compounds which are in potatoes into the smaller sugars. And then it can travel around the body.
So that's all in our mouth, in our saliva, and that sort of thing?
Yes, and then there is in the stomach. Liver is absolutely full of different kinds of enzymes because liver is basically helping us to keep safe and healthy, and it's a cleaning organ of our body.
Okay, now I've noticed you've got some beer with you as well.
I do.
So tell me, what does this have to do with catalysts?
Well, usually, beer is made of cereal, and barley is one of them. And barley has lots of different compounds which need to be degraded into the smaller elements, and also alcohol, because beer contains alcohol and different aromas. And to help us to do this, we usually take yeast. Yeast has lots of catalysts, lots of enzymes to do that.
And that just speeds up that reaction.
Speeds up the reaction and makes it go much faster than usual. And I have a very special beer here made by an artist, and it doesn't use yeast, but it uses saliva. So she used, this is an artistic project. She claims that there will be a character that you can feel in this beer. And basically the enzymes for saliva are used to transform the compounds into the alcohol. And I think we should try some.
Okay, we're going to do this. So instead of having yeast, we've got saliva to ferment this beer. Okay,
yes.
Let's give it a go.
And so this is a beer with a character. So let's try some. Okay.
The beer's just sort of gone everywhere.
No, this is really good. The character is all over the place.
I mean, that's certainly one way of putting it.
Go on then, pass them over here. It's quite cloudy, isn't it?
It looks more like cloudy lemonade.
Yeah.
Yes, it is cloudy because it has lots of small compounds which are transformed, with the help of the catalyst. And so it's very organic as well.
It smells interesting.
Here we go, Katie. Cheers.
Who's going to be the bravest?
Cheers, Liliana.
Cheers.
I think it'll be Izzy.
Oh, gosh.
You see, it's kind of refreshing.
I mean, that's certainly one way of putting it. I wouldn't say it's a beer, essentially. It doesn't taste like beer.
Yes, exactly. I mean, beers can have a different aromas as well. So this would be a little bit lighter, although it's a Brazilian donor.
I mean, how has this actually been made? How do you get a few products? Does someone just spat into a bottle? Like, what's going on?
Well, I mean, the artist that made this, Clarissa, the beer, has used a similar principle to how you would basically brew anything else. So she used cassava as the main ingredient. For beer, we will use barley or any kind of cereal. And then she added a little bit of sugar to it. If we use a good barley, we don't necessarily need sugars because there are already some kind of sugars in it. And then she added saliva. And saliva is full of different enzymes that can cleave the cassava ingredients into the smaller pieces. And one of the smaller pieces will be also alcohol. And there will be a little bit of aroma, and there will be also a little bit of bubbles. You notice?
Yeah, I did notice that. It's fizzing away.
Yeah, so bubbles are CO2 or carbon dioxide, which is also produced in the reaction.
Okay, so where has this saliva come from?
All of this molecules are important for our psychology. So they determine our mentality, our character, because they regulate production of hormones in our body. That means they control a little bit our feelings. So she got 4 volunteers to give the saliva test. So you can have 4 different beers. Currently, this is the only bottle in UK.
Oh, so we're very privileged, I think,
yes.
to be trying this. Well, Liliana, thank you so much for joining us today. That was Liliana Fruck from Cambridge University.
I'm sorry, I've got to admit, I was a chicken and didn't try that beer. I think I might need a bit more convincing.
I can't believe you just left me to drink that on my own.
So those are nature's catalysts. But one of the most commonly recognised applications of catalysts are catalytic converters found in your cars. And Tamsin Bell used hers on a little road trip.
Have you ever thought about what comes out of your car? We often hear about the nasty effect car pollution has on the environment. But is there anything we can do to reduce it? I went to Johnson Matthey, a company that makes devices called catalytic converters that do just that.
Yeah, hello, my name's Chris Morgan. I'm the Technology Director for Johnson Matthey, where we develop catalytic converters.
How much of an issue is air pollution?
Particularly in cities where there are high concentrations of particulate matter, soot and oxides of nitrogen in the air, that can cause problems with people with respiratory diseases and asthma. So it's important we try and clean those emissions up to improve people's health.
So what comes out of our cars?
You inject a mixture of fuel and air into the engine, and that doesn't burn completely cleanly. So you get unburnt hydrocarbons from the fuel, you get carbon monoxide, and with any high temperature combustion process, you get oxides of nitrogen formed, as well as soot from both gasoline and diesel vehicles. The catalytic converter is important in terms of reducing that pollution to lower levels and improving the health of everyone in those urban environments.
And how can you test for these things?
We make development catalysts and we fit them onto vehicles, onto engines, and we measure how effective they are.
And can we go and see this?
Yes, I'll take you to the lab now.
Johnson Matthey are pioneers in developing chemical technologies to make our air cleaner, using a device called a catalytic converter to transform the polluting particles from our cars. But what would our cars release without these devices? I went to the lab to find out. Chris showed me a control room at Johnson Matthey's Technology Centre, a room stacked with analysing machines that overlooked the testing lab.
Yes, so if you look through the window here, we've got a passenger car sitting on a rolling road. So there are big metal rollers underneath the ground that the vehicle sits on. And when the test starts, those rollers will turn round and that will simulate the vehicle driving along the road. We have a big fan that sits in front of the car to help keep it cool while it's running the test, otherwise it would overheat. And then at the rear of the car, we're collecting all of the exhaust and taking it through to these analysers behind us where we can measure the concentrations of the pollutants that are being generated.
As the test started, an array of numbers flooded onto the screen.
We are measuring what's coming out of the engine, and you can see different gases. We're measuring hydrocarbon, carbon monoxide, oxides of nitrogen, and quite high levels. You can see coming out of the engine we have thousands of parts per million hydrocarbons.
Essentially, there are thousands of polluting molecules coming out of our car all the time. But could a catalytic converter change all that?
This catalytic converter is based around a ceramic honeycomb, which sits within the exhaust of the vehicle and has thousands of small channels running through it. And what we do at Johnson Matthey is we apply coatings onto these channels that help to reduce the pollutants that are passing through the unit.
So you're holding one of these catalytic converters now. So this is like a cylinder and it's about 15 centimetres in diameter and about 20 centimetres long. It looks quite big. Is this the normal size?
For a passenger car, yes. Typically, the size of the catalyst relates to the displacement of the engine. Wherever possible, we get it as close to the engine as we can. So if you look in the engine compartment, they're often located just downstream of the exhaust manifold that comes out of the engine.
So we're looking at the screen here and we can see lots of horrible things coming out without a catalytic converter. But what happens when we do have the catalytic converter?
Yeah, so if you look at these two sets of numbers, what's coming out of the engine, we've got hundreds or thousands of parts per million of these pollutants, the hydrocarbon, and then we measure at the end of the exhaust system behind the catalyst, then that number is down into single figures, so we're converting a very high proportion of the pollutants over the catalyst.
You heard right. A catalytic converter can take over 1,000 molecules that are harmful to the environment and reduce them to just 1 or 2. But how?
Within this honeycomb, the coating that we put on contains precious metals, so typically platinum, palladium and rhodium, and those form millions of tiny islands within the coating, And when the pollutants land on those precious metals, that helps to react them with oxygen and convert them to carbon dioxide and water.
And why do you use this honeycomb structure?
Because you need to have the maximum interaction between the gas and the active component. So by having these thousands of channels and the coating applied in those channels, there's a much shorter distance for the gas to travel to hit the active part of the catalyst and for the reaction to take place. If you just coated the outside of the pipe, a lot of the gas would go down the middle and never touch the catalyst.
How would you know if you need to get your catalytic converter changed?
Well, hopefully you should never need to get it changed. They're designed to last for the lifetime of the vehicle. The legislation says they should last at least 10 years, 160,000 kilometres of driving. But all modern vehicles have a diagnosis system that monitors how well the catalyst is working, and if it detects a problem, then you get a warning light on the dashboard and you need to go to the garage and get it checked.
And what next? What can we improve on?
The big change that's happening at the moment is not just testing the catalysts in the laboratory under defined test cycles, but making sure that they work under a much wider range of conditions out in the real world. So we're now not just testing catalysts on vehicles in the laboratory, we're fitting them to cars with a portable analyzer system strapped onto the back of the car, and we're driving out in the real world in winter, in summer, seeing real traffic conditions and making sure the catalysts work well under all of those driving styles.
That was Chris Morgan there from Johnson Matthey and Tamsin Bell.
Now, we've heard how catalysts can help to clean up what's coming out of our cars, and using metals to speed up reactions has also revolutionised the way we make things on an industrial scale, from plastics to fertilisers. Without them, it would take months or even years to get your end product.
And one example of this is the production of synthetic liquid fuels. We currently use petrol or diesel in our cars, and these come from natural oil reserves. But burning these releases a lot of harmful chemicals, as we heard earlier, and they could one day run out. Instead, we can use catalysts to artificially make cleaner fuel to power our cars or even aeroplanes without releasing as many nasty gases. Katie spoke to Professor Robert Tooze, who's an expert in synthetic liquid fuels and currently directs Drochid Research Services in St Andrews.
The majority of fuels that are used now to power vehicles come from oil through oil refining. So this process is breaking down large molecules into the products that our cars run on. So a synthetic liquid fuel is a hydrocarbon fuel similar to conventional petrol and diesel, but it's not derived directly from oil. So this is not a new process. The original discovery by two German chemists called Fischer and Tropsch goes back nearly 100 years. This originally converted coal via a multi-step process into liquid fuels like petrol and diesel. This was later commercialised in Germany and then more globally, both in South Africa and beyond. So this is a quite remarkable process for converting coal into petrol and diesel. More at a molecular level, the way you think about this, rather than oil refining, which breaks up large molecules into the smaller molecules our engines burn, this goes in the opposite direction. So it takes very small molecules and stitches them together to give you the right structure for liquid fuels.
And why is it better to make fuels from coal, say, than current methods of getting petrol?
If you look back in history to the synthetic liquid fuels, it's more about necessity being the mother of invention. So in both of the regions where this technology was first pioneered, there were ample coal deposits but no oil. So the conventional routes weren't open to them. So they found a way to convert coal into petrol and diesel. The attraction of these synthetic fuels is they've got inherently low sulfur. So sulfur is one of the key contaminants that cause environmental issues. The other thing that we're increasingly aware of are particulate formation by engines. These synthetic fuels are very low in the compounds which lead to the formation of particulates.
Step by step, can you take us through the process of what happens? Say I've got my coal or my biomass or my natural gas as my carbon source. How do I get petrol out the other end?
There are three main steps in the synthetic fuel process. The first one is taking your carbon source, whatever that is, and converting it into the correct mixture of carbon monoxide and hydrogen called synthesis gas. The second step is really the catalytic step. The carbon monoxide and the hydrogen is passed over a catalyst at elevated temperature and pressure to be stitched together to give you the molecules that you need as the fuel. In a third step, you finally do some product treatment to give you the diesel and petrol that car engines require.
So why do we need this catalyst for the second stage?
A catalyst speeds up the rate of a chemical reaction. So if you didn't add a catalyst, the reactions would require massively high temperatures or very long times, neither of which would make this commercially viable. The other thing the catalyst does, it controls the selectivity. So it gives you some control over the products that are made. And again, what that means is it's both energetically and materially efficient. So you get from A to B in the most cost-effective and environmentally sound way.
Oh, I see. So if you put the carbon monoxide and hydrogen together in a room, they might not necessarily do much, but the catalyst, well, catalyses the reaction.
Exactly. You would wait a long, long time for carbon monoxide and hydrogen to react if you just left them alone.
What typically is the catalyst used in this reaction?
Typical catalysts are based on cobalt or iron.
And how much do these sorts of materials cost?
That varies very much from process to process. Their cost contribution to the overall process will be very low and would have to be economically viable. So even though some of them are based on very precious metals, if you get an efficient enough catalyst, then the actual cost is low. So one of the key defining factors about a catalyst is its so-called lifetime. So how long it will perform at the level you want.
And is this still being used today?
It is still being used. The beauty of a synthetic fuel process is it doesn't really matter the original form of carbon that you feed to this process. So as I say, the historical background was using coal, but you can also use natural gas. And in the future, it's possible to use other raw materials, so waste biomass as the source of carbon to make synthetic fuels.
So household food waste could be your carbon source for this process?
Yeah, potentially that is a way. Anything that has carbon in can eventually be turned into synthesis gas. The question about the economic viability of that's critically important. Too expensive, they won't get taken up by consumers and therefore they won't have any environmental benefit.
Given the concern over climate change, even the recent IPCC report, could carbon dioxide itself be used as a source for this process?
Potentially, yes. There are many people working on the conversion of carbon dioxide into liquid fuels, and that's a challenging catalytic problem.
Do you think the future of these synthetic liquid fuels is quite promising?
We have to recognise that we're in a transition period for energy. We increasingly see electric vehicles, and their growth is rapid, but the number of electric vehicles is small. So we need to manage the transition to some future beyond hydrocarbon fuels. And what we can do in the meantime is to make the cleanest fuels we can. I think synthetic fuels could find application where other energy vectors are difficult to see. So, for instance, air travel. People are talking about electric planes, but for the foreseeable future, they're going to run on liquid fuels. So there's an opportunity there to make sustainable aviation fuels. And catalysts are important both currently and perhaps increasingly going forward.
It would be pretty amazing if we could take that carbon dioxide from our cars and use it to make more fuel. That was Robert Toos from Drochid Research Services.
So, talking of environmental pollution, as we look forward to the next generation of catalysts, could these help us to clean up our environment? Carbon-fluorine bonds are some of the strongest in the world, and they're found in products that we use every day. But the problem with these strong bonds is that, because they're really strong, we can't break them down. And so they can build up in the environment, and some of the chemicals that contain these bonds are harmful. Recently, a group of researchers in the US have managed to make a new type of catalyst that could break down these carbon-fluorine components in materials and pollutants building up all around us. Dr. Eli Stavitsky, who is involved in the project, is on the line to tell us more. Now, Eli, where can we find these carbon-fluorine bonds?
You might remember the ozone hole scare dating back to the 80s. The primary cause of this was so-called chlorofluorocarbons that we used in refrigerators and air conditioners. They were eventually banned and replaced by other materials containing fluorine. which are less harmful to the ozone layer, yet they're many times more potent as greenhouse gases compared to carbon dioxide. The specific chemical we targeted in our work is used worldwide in coatings, and the current contamination levels are truly troubling, and that, together with the carbon-fluorine bond being one of the strongest, really made us look for new ways to clean up this mess.
Okay, and what is the chemical that you've been looking at? Tell us a bit about it.
It's a perfluorooctanoic acid. This is a long chain of carbon atoms all connected to the fluorine bonds used in many products that we encounter every day. In the process of preparing them, there is a contamination arising from the process.
So what kind of products is it in?
This is used in firefighters' foams for the coatings on your pans and skillets, in the coatings that protect different products, so it's really widely used.
So do we have any reason to be worried about the non-stick pans then?
Oh, no. The pollution occurs in the process of their preparation.
Oh, I see. So can they be harmful in any other ways?
The materials that contain carbon and fluorine, such as perfluorooctanoic acids and related compounds, are suspected at the moment to cause cancer and reproductive effects. And this is why the agencies around the world are looking at them to determine how harmful they are.
So there could be potential health impacts as well as environmental impacts. And crucially, these bonds can't easily be broken. So these compounds are sticking around for a long time, wherever they end up. Now, you've been trying to tackle this problem, haven't you? So tell us about these catalysts, these so-called single atom catalysts.
Yes, we managed to find the means to break this carbon-fluorine bonds using single atom catalysts. Ultimately, you want your catalyst particles to be very small, and this is because the catalytic reactions occur on the surface of the catalyst. So you can make nanoparticles out of them, but ultimately you can go one step further and break them into single atoms. Breaking large pieces of catalyst brings about very unique properties. Think about a golden ring on your finger. It's as chemically inert as it goes, but when it's broken into the pieces of a billionth of a meter, it will turn over many chemical reactions at very high rates. The ultimate goal here is to arrive to a single atom, because that's how we will optimize our catalytic activity.
So you can take a metal that on a macro scale would be pretty inert, and by changing the scale in which you're operating, the properties change.
That's exactly correct. And the ability to break the catalyst into single atom brings about even more new functionalities and new useful properties.
What material are we using here?
In this work, we're using platinum, which is a precious metal. Platinum breaks hydrogen molecules into atoms that are very, very active, and those atoms are those that cleave the carbon-fluorine bond. But ultimately, there are many people on this planet right now working on single atom catalysts of all different metals.
So looking forwards to the future then, what could these single atom catalysts do for us?
Oh, there are many processes, many emerging reactions where single atom catalysts show amazing activity. But the catalyst we developed, it still contains precious metals, and we really have to look to find the useful functions of abundant, less expensive materials such as iron, cobalt, and such. But ultimately, single atoms are not the final goal. We're just starting to manipulate single atoms of different kinds, arranging them in ensembles of 2, 3, or maybe more, where each atom will perform its own unique function.
Wow, so it sounds like there's a lot of potential then and a lot of work that still needs to be done.
Oh yeah, this field is in its infancy, and I'm so looking forward to be a part of it.
Eli Stavitsky, thank you very much. We'll have to end it there. Eli Stavitsky from Brookhaven National Lab. And thanks to all our other guests this week, Chris Morgan, Liliana Fruck and Robert Toos. Well, Izzy, I'm convinced next time I hear the word catalyst, I won't have troubling flashbacks to that confusing chemistry lesson at school.
Oh, well, I am glad to hear it. Well, we've just about got time for Question of the Week, and Tamsin Bell's been lighting up on this question from Bethany.
My colleagues and I have had a rather heated debate in the office recently, sparked by one of them saying that second-hand smoke is more likely to give you cancer than actually smoking a cigarette. I was shocked when I heard this and struggled to believe it. What do you guys reckon? Is she right?
In several countries around the world, new legislation means that smokers see a graphic health warning on cigarette packaging as a constant reminder of potential side effects of smoking. But are the non-smokers at risk of these too? We asked Stefan Marciniak, Professor of Respiratory Science at the University of Cambridge.
This is a good question, and as with so many good questions, the answer isn't straightforward. It's both yes and no. Passive smoking, also called second-hand smoking, is recognised by the World Health Organization as a major global health problem. Every year, nearly 7 million people around the world will die of smoking-related illnesses. 6 million of these are smokers, because half of all smokers will die of a smoking-related disease such as cancer, heart attacks, strokes or emphysema. But the remaining 1 million deaths are in non-smokers. That means that roughly 1 in 7 smoke-induced deaths is due to second-hand smoke. Cigarette smoke contains more than 4,000 different chemicals. Of these, we know for certain that at least 250 are toxic and at least 50 can cause cancer, such as arsenic, lead, and cadmium. There is no safe limit to cigarette smoke exposure. Cigarette smoke comes in 2 forms: mainstream smoke, which is the smoke drawn into the mouth of a smoker, and side stream smoke, which is the smoke coming off the end of a smouldering cigarette. Second-hand smoke is a mixture of exhaled mainstream smoke mixed in with side stream smoke. In a sense, it is true that side stream smoke, often experienced by bystanders, is worse because it contains much more carbon monoxide and other toxic chemicals such as ammonia and nitrosamine compared to mainstream smoke because it hasn't gone through the filter at the bottom of the cigarette. However, a passive smoker will be inhaling more dilute smoke compared to an actual smoker, so the smoker will still receive a larger dose of the toxins, and in that sense, the passive smoker isn't actually coming off worse. But we mustn't underplay the dangers of passive smoking. Globally, about 1/3 of the deaths each year caused by second-hand smoke are in children. In the UK, passive smoking causes 165,000 new cases of disease in children each year, including asthma, pneumonia, and tragically even sudden infant death syndrome. Even the unborn child is at risk. Pregnant women who are exposed to second-hand smoke are more likely to have low birth weight babies, and these babies are more likely to be born prematurely. This is why since October 2015 it has been an offence to smoke in a car with a child, and it's also the reason why smoking isn't permitted in workplaces or social spots like bars and pubs. So to summarise, if possible, you should try to stay away from any type of cigarette smoke.
Thank you, Stefan, for such an informative answer. It really highlights the importance of not hanging out in the smoking area to look cool. Next time, we're sniffing out this more light-hearted question from Andrew.
How do wombats produce cube-shaped droppings?
I cannot wait to hear the answer for that one. If you have any guesses, why not email chris@thenakedscientist.com, find us on Facebook, tweet @nakedscientist, or join in on the debate on our forum, thenakedscientist.com/forum.
And that's just about all we've got time for this week. Thanks to Tamsin Bell for putting the programme together. Do join us next time when we're hanging up our lab coats and slipping on our dancing shoes to explore the science of having a boogie. The Naked Scientist comes to you from Cambridge University and is supported by the EPSRC and Rolls-Royce. I'm Katie Haylor. Thank you for listening. Until next time, goodbye.